louder, sorry.
It's 2 to 13.
That's right. That's what I said. I mean, if you have a hard time with that and you need an extension or something, email me, but it should be. These are, suggestions. I'm much more concerned about you doing the reading and interacting with each other than I am with when you get it done exactly. Though, there's a lot to be said for reading the material before you take a test on it. Other questions like that. So let's catch up to where we are. We've left C. elegans behind because that mosaic specification of cell fate just doesn't work when you have billions of cells you got to make. Instead, what we've seen is there's a conditional specification of cell fate. And to some extent, it's not just conditional, it's also positional. What fate you take on depends on where you happen to find yourself. Okay? And so the first and most important thing is that the egg is not homogeneous and that there are differences between it, within it, that we're going to end up, that the blastula eventually ends up with more beta-catenin in one particular part of the egg, and that mom arranged for beta-catenin to induce the dorsal lip of the blastopore to start making organizers, and then the dorsal lip of the blastopore, as it does gastrulation and that goes under into the organism, you'll have a layer of mesoderm that is inducing the ectoderm above it to make a nervous system. And so everything's hunky-dory, right? It's as simple as that. You gotta thank mom because she had to put the beta-catenin here. But otherwise, with the organizer getting things started, you start making an entire nervous system. And you can at least conceptualize, we're not going to talk about the details, you can at least conceptualize how once you have an anchor point and something gets started, that all the other cells in the organism, all they have to do is say, who's around here? Oh, I'm going to do what they're going to do. Or I'm going to do at least what's appropriate for what they're going to do. And in doing that, you can end up with an organism with trillions of cells. And yet every cell is the appropriate kind of cell for where it is. And by being the appropriate kind of cell, I mean it is expressing the genes that are appropriate for a cell in that place that needs to do that job to take on that structure and to start fulfilling that function. So it's all a matter of controlling gene expression. And it's not just what's the cell expressing now. We're going to see more examples of this in chapter 2. Sometimes it's a question of what has been the sequence of gene expression that particular cell went through. And crucially, many of those genes being expressed are themselves transcription factors. And so they are constantly toying with the genome to get a particular cell down a particular pathway so it will be a very nice motor neuron in your spinal cords, or a very nice liver cell in your liver, or a very nice tonsil cell, whatever it is tonsils do. So you can at least conceptualize how that would work. Okay? Questions about any of that? Yeah, good.
Is the dorsal lip, does that become the notochord? Or
it's a great question. Yeah, the dorsal lip of the blastopore, as it goes in and becomes mesoderm, some cells in the middle are going to coalesce at the midline and form the notochord. Exactly. Justin. Okay. And I'm going to switch topics a little bit, unless there are more questions about that. And as Justin says, it's the notochord that's going to be releasing, right, a blockade of the bone morphogenic protein, and therefore this part of the of the neural tube is going to end up having a different fate than that part of the neural tube, and we'll see that later. Okay, so I always want to talk a little bit about what we know about development of the insect nervous system. And the first thing we have to deal with when you're dealing with the insect nervous system is while your nervous system, your central nervous system, is along your dorsal side, all the invertebrates, and that's typical of vertebrates, all the invertebrates, they all have their nervous system on their belly side. That's weird. Why was that? Don't we have a common ancestor? Indeed, we must, right? And so, but I want to point out to you that there's a bit of terminology that you might not have thought of before. So belly, ventral, back, dorsal, which is those terms are really put in place by the mouth. Where is the mouth, right? My mouth happens to come out this end, yours too, I bet. And we call that end the ventral surface. And the other side is a dorsal surface. But maybe mouths don't have to always be in the same place. So the typical insect nervous system looks like this, where there is a string of what are called ganglia that are along the ventral surface. And when we talk about segmentation, you'll see more of this later, that each insect segment has its own little ganglion in the ventral surface. And the way to understand the insect brain is that it's a part of the animal where several segments came together, squished together, and shoved all the ganglia together to make a much more complex neural system. And so we'll call it a brain. And the brain is connected by axons to the rest of the nervous system. All those ganglia, there's a chain of ganglia going down. And there are connectives between the various ganglia, and the ganglia send out axons to control muscles in that particular segment and to receive sensory information in that particular segment. So it's all very nicely organized. And although this diagram doesn't show it, within each ganglia, there are some axons that cross the midline to talk to each other. No surprise. This business of it being on the belly, there's one thing that may look a little bit awkward to you. You see this gap here? That gap is there because that's where the animal's mouth is. Food has to go through there. So you can think of these ganglia in the front, they got squished together and just in front of the mouth, and then the mouth went here, and so all the connectives have to work their way around the mouth. Sounds like so don't talk to me about how well-designed creatures are, right? This is a dumb idea. It's almost as dumb as breathing and drinking in the same tube, right? But we're stuck with it. Natural selection hit on that, and our ancestor kept making descendants, and so we're stuck with it. Questions about that? So how did that happen? This difference. And a long time ago, before Darwin, there was a French scientist who made a suggestion. And I need to point out to you that before Darwin's theory of natural selection, people already had the idea that animals evolve from one species to another, or that some species may have evolved from the idea of evolution. That wasn't revolutionary with Darwin. In fact, Darwin's grandfather was famous for having written a book about how various animals could have come from, could have evolved, changed over time. It's just nobody had a mechanism for how that happened. That's what Darwin's theory of natural selection provided, right? That then got reinforced by Mendel's discovery that adaptive traits could be inherited discretely as a whole in at least some offspring. So people were already thinking, how did this happen? That the invertebrates have the nervous system on the ventral surface and we have ours on the dorsal surface. And so this guy pointed out what I just pointed out to you is what we call dorsal and ventral. That's just a matter of where the mouth is. And so he proposed two ways that the change could have happened. Presumably the ancestor was a invertebrate type, right, with the ventral nervous system. And what if at some point in the common ancestor of all the vertebrates, what if evolution arranged for the mouth to come out the other side? Instead of coming out on this side, to come out that side. And then when we look at the creature, we say, oh, this is the ventral surface and that's the dorsal surface. But in terms of building an organism, the nervous system doesn't have to do anything, right? It just stays right where it is. And you put the mouth the other end. And then we humans thinking that, when we apply words to something, we've explained it. we say, oh, that's the ventral surface, never being the wiser that that used to be the dorsal surface in our common ancestor. And so that sort of neatly explains how that could have happened. But he also suggested another thing that could have happened in evolution, which is not just that instead of the mouth moving, maybe the head rotated 180 degrees. And in rotating 180 degrees, it would also rotate the mouth. So now what used to be, right, our back now has the mouth going through it, and now we're calling it the ventral surface. What's interesting about that solution is that it also explains an organization in vertebrates that's not seen in any invertebrate. If we go back to our insect here, this part of the nervous system controls that part of the body and vice versa. There's no crossing. I mean, there's communication between the two sides, but the left side of the nervous system controls the left side of the body and vice versa. And as you know, in invertebrates, that's not the case. And so he suggested that if what happened is that the head turned, the whole head turned, then the nervous system would turn too, and now you'd end up with the left side of the brain controlling the right side of the body and vice versa. Everyone see the logic of that? Don't ask me how you have evolution outer ranges that twist the head around. Maybe having the mouth on that side was important for some reason, and it hit upon that version of it. So what's cool is this would explain this thing that every neuroscience student has to learn, which in us vertebrates, there's this crossover. And by the way, in some places, like here in the brainstem, there would be axons that would actually cross the midline, as in the pyramid and in the medulla, right? Where there's this decussation across the midline. Everyone got that? Now, how many of you have heard of this idea before? Oh, excellent. I mean, and not from the book. Okay, yeah. What's interesting about this is it explains that business left control of the right body and all that. Have any of you heard any? Yeah, no, Abdur.
Question. The scientists suggesting that the whole 180-degree turn happened in one gen?
No, clearly not. And if that seems weird to you, that might be, well, let me point out to you, how many of you have heard, how many of you like to eat sole or halibut? Right? Fish, right? These are fish who start off with their head like every other fish. And then as they grow older, one eye rotates around and their mouth ends up down here so that they're bottom feeders. Right? And clearly that works, right? So structures on the head can move around, right? We know that. There was something, yeah, Laura?
I don't know what this said head rotating has to do with the mouth going to.
Well, if have you seen The Exorcist?
Right.
So if I were to rotate my head 180 degrees, my mouth would now be facing that way, not this way.
Like,
and you wouldn't be able to hear me because the microphone wouldn't be the right place.
why didn't you just use the first model? Like,
you can, but it doesn't explain why the left side of the brain controls the right side of the body.
but I feel like in evolutionary terms, that's more complicated than to just go with that.
I agree with you entirely. So instead, we have to have the alternative explanation for why the left side of the body controls the right side of the left side of the brain controls the right side of the body. What is that alternative explanation? I don't know of one. I've never heard of one. There is none. So you got to pick your poison. Do you want a complicated change in the common ancestor to the vertebrates? And then you got to come up with some other mechanism to explain this decussation of all the control? Or do you go with this explanation and say, oh, at least I've explained two things at once. I've explained why our nervous system is on what we call the dorsal surface and why we have this weird thing going on in our brain. And if you look at the invertebrates, it doesn't have to be that. There's nothing about having a body and moving it around that requires there be some crossover of axons in the neuraxis, but it happens. Okay. Think about that some more. Okay, given that in insects, the nervous system is going to appear on the ventral surface as opposed to vertebrates where it appears on the dorsal surface. But the whole business of the organizer, that is a mesodermal tissue, releasing something to cause the overlying ectoderm to make neurons, that's exactly the same in insects. So that was already established in our common ancestor with the insects, right? That the mesoderm would tell the ectoderm to make a nervous system. The details are different. For one thing, chordin in vertebrates, its evolutionary precursor is called SOG in fly. But it serves the same purpose. It inhibits the equivalent of bone morphogenetic protein signaling in transforming growth factor beta receptors. They got different names because when they were first found in insects, they got a different name, but it's the same mechanism, the very same. And the other the other thing that's going to happen is that it will be some part is far away from the signal and therefore will make skin, and other parts are close to the signal and therefore are going to make nervous system. And this gets started at the stage where the embryo may only be one cell thick. And so you reach a stage in insect development where it consists of, a one cell thick balloon, think of it that way. And at one side, what happens is some cells get out of there, leave the single layer, and start migrating up to form the nervous system. So if we look at a developing grasshopper, for example, we can keep track of genes that are known as proneural genes. They're called proneural genes because any cell that's going to become a neuron has to express these genes first. And they have funny names, a Kate Cute, named after the fact that when they isolated the genes in flies, it was because they caused, interesting changes in the morphology of hairs and skin stuff. So unimportant for us. Its real function, even though it has consequences for hair, right? Pleiotropy strikes again. Its real function is to start genes along the pathway of developing into neurons, start expressing the right genes so that they will become a neuron. And if you look at the ventrum of a grasshopper embryo, you can keep track of where the Kate-Cute genes are being expressed. And lo and behold, they're in these little rosettes, one clump on each side, and there's exactly one pair of clumps per segment. These cells are going to eventually make the neurons that make the ganglion on that side in that segment. Now, there's a question. How come these cells are where they are laid out? Look how regularly they're laid out, right? What put them there? And I don't know. I'm not sure anyone knows. But presumably something induced them to start expressing a Kate-Cute. And whatever that inductive factor is, it too is distributed so that you end up with this nice lattice-like expression pattern. Good. Maria?
Or that's just like, have you tried that?
The rosettes at this stage, it is a clump of cells in the outside, in the epithelium, who have begun expressing a Kate-Cute. And the question is, what happens next? And I don't, I'm not offering an explanation of why the rosettes form, but within each rosette, they're eventually going to give rise to a ganglion. And what happens is very interesting, which is, here's a rosette in cross-section, and what happens is, as development proceeds, one cell, typically the cell in the middle of the rosette, one cell starts rising up to enter the body cavity up here. And as it rises up, it starts expressing a Kate-Cute more and more strongly, and all the neighbors who get left behind express it more weakly. So that at a later stage of development than this, you don't see rosettes expressing a Kate-Cute, you see one cell expressing a Kate skewt in each of the sides of each of the ganglia. This process of leaving the layer is known as delamination. So within each rosette, one cell delaminates, and that cell is going to give rise to all of the cells in that side of the ganglia, all the neurons, glia, etc. So it rises up from the, and it becomes, because it's going to divide many times and make lots of neural things, it's called a neuroblast. And it's different than these skin cells who are never going to express a Kate skewt again and are never going to become neurons. They're going to be skin cells. So what I always think of is that scene in Toy Story where the crane reaches down to grab Buzz and pulls him out from all the guys who are the same, and they say, he has been chosen, right? One cell is going to pull up from the rest, and it's going to be different from them from this point on. And so you might ask yourself, well, how did that happen? And you never see two cells delaminate, and you never see three cells delaminate, and in every rosette, there's always a cell that delaminates. That by itself tells you that there's got to be some sort of communication going on within the rosette. Because, I mean, it's just a probabilistic thing where you've got a 55% chance, you've got a 30% chance, right? Then there'd be some ganglia where nobody would delaminate and others were more than one, and yet you don't see that. So they have to be communicating with each other. And again, the fruit fly people worked out the details of that, and it's very interesting for thinking about neural development generally. So, number one, this is still conditional specification of cell fate. Because what you can do is you can wait for one cell to delaminate, and you know that normally that would become a neuroblast. And you run your laser through the microscope and you kill that cell. So is the animal going to do without a ganglion on that side, in that segment? Nope. Because as soon as you kill that cell, these cells that had stopped expressing a Kate skewt, they start expressing it again. And so you make a new rosette that's expressing the proneural genes, and eventually one of them delaminates, becomes a neuroblast, and will populate the ganglion on that side. Good. Yeah. Say it again.
There's the pick location within the rosette.
You know, typically it ends up being in the middle of the rosette. And maybe not so coincidentally, the strongest expression of a Kate skewt often seems to be in the cell that's in the middle of the rosette. Presumably that means they all got some induction factor that told them start expressing a Kate skewt, and maybe the one in the middle got the best dose of it, highest dose of that. But what happens next is they fight it out to decide who's going to really become dominant. And we understand the molecular basis of that. Laura?
We don't know, like, who puts a pro-neurogen in there.
Well, I don't know. I mean, I don't know if people know, but presumably it was some earlier inductive process. Yeah, that's fine. Amy?
That's my thing. For new delaminated cell guys, does a new rosette form, or does one of the side, like?
Well, a new rosette forms, and it tends to be right where the neuroblast you just killed was, and they start going through the whole process over again. And yes, you can kill the next neuroblast, and another one will come along. Well, I don't know they ever took it past 2, but it seemed to be there.
Yeah, I just wondered if they were able to stop communication between the two different genes they cells.
Eventually, they were not here, but and they did it the way, you know, fruit fly people, they don't do surgery and stuff. This business of having laser and actually manipulating a fruit fly embryo, that's kind of the exception. For the most part, in fruit flies, you make manipulations by making mutations. And through using various mutations, et cetera, they establish the molecular network that's involved. And that's the so-called notch delta system. In each rosette, every cell because of the Kate skewed, the proneural genes, they're all expressing two genes, Notch and delta. And these are cells that they insert into their membrane. And this is a system whereby one cell can come to dominate over the other. And this happens because one cell is affecting the gene expression of another, right? That's the only sort of communication that matters for cell fate, is this cell is telling that cell which genes to express. So they're both membrane-bound proteins, and delta binds to Notch extracellularly. When that happens, when delta binds to Notch, then there's one intracellular portion of the Notch protein that breaks off. And it then enters the nucleus and, interacting with another protein called enhancer of split, they start suppressing a Kate skewed expression. So they're going to be pushing this cell to stop going down the neural pathway and go back to being an epidermal cell. And one of the things that happens when you suppress the Kate skewed is this cell doesn't make as much delta anymore. So one cell is telling the other cell, you stop expressing delta. I'm the one that's going to express delta around here. And if you're touching me, my cell is going to tell you to stop. My membrane is going to tell you to stop making that. Notice this is a positive feedback loop. If one cell happens to have slightly more delta expression than its neighbors, then it's like the American system. The rich get richer, right? It will then suppress any further expression of delta from its neighbors and eventually become the only one that's expressing delta. And once that happens, it delaminates and goes down the pathway of making a ganglion, of dividing many times to make lots of cells, all of which came from that one neuroblast that delaminated. And as I'm trying to express it here, if this cell happens to be expressing the most Kate skewed in response to some inductive factor we're not going to talk about, then that means it will be expressing the most delta. And once it starts expressing delta, it will be suppressing expression of delta in its neighbors. And notice this is a winner-take-all system. Only one cell can win. How's that? Good.
So the Stalwarts dice, is there anything different about it?
Many things are different about it. Yes?
Like to make it express more?
Absolutely. So there's a positive feedback loop there too. We're not going to talk about it, but what's going to happen now is this cell is going to go down a cascade of gene expression, including many transcription factors. They're going to push it more and more to express the sort of gene they need to be a neuroblast. And as each cell divides in the neuroblast and forms its own, it turns out, I don't know if I have this here yet, but which fate those genes take on, that'll be next week. Which fate each cell, the first cell budding off of the neuroblast, it's going to have a different fate than the second cell, which is a different fate than the third cell, etc. And so their specification is based on, what was being expressed in the neuroblast at that time. And there's, Laura.
They can't all be the same, and I think at least one of them has more delta protein than the others, right?
Perfect. At least one had to have a little bit more delta protein than the other. Variation happens, right? This is the mantra of biology. Variation happens. Yeah.
So in that diagram, I understand why the central protein has the most delta protein, and then it outcompetes the purple cell.
Yep.
Why wouldn't it be more favorable to have the corner cell of the rosette turn into the neuroblast? Because then it would have minimal inhibition from the surrounding cell.
So I mean, I get what you mean. If the rosette was big enough, then you could imagine there might be two winners. And so apparently the rosettes just the right size so that there'll only be one winner. And it's not a coincidence that the winner tends to be the cell that was in the middle, i.e. got the most inductive influence, made the most acute, and had a little bit of a head start. Likewise, right? In the American system, if your dad gives you $80 million, you're more likely to be a success than if he doesn't. That's just how we do it.
So you said that in biology, variation happens.
Absolutely.
It has to happen because otherwise the individuals not have a nervous system. So you kind of like counting with variation.
Well, and let me say, there are times when the system doesn't work, and those flies leave no offspring because they're going to die, right? So that sort of variation happens too, right? As we talk about in human sex, there are plenty of embryos that get started that mom never even knows they were there. Something went wrong because variation happens. And Darwin didn't say every population would be full of uber individuals, right? He said every population, varies a lot and how adaptive and fit they are. Some produce a lot of offspring and some produce very few.
So it kind of like depends on their niche.
And exactly. If every, pick a species, if every chimpanzee was absolutely perfect in every way, well then, there'd never be any change. And had that happened to us, I guess we'd still be some common ancestor to chimpanzees. But change happens because variation happens. Because natural selection has to work on variation. Okay. And this is just another, I'll just say, the preview here, this is another way I tried to have the artist visualize this. And so take a look at it. But I will say that when we get to the vertebrate nervous system, we'll find that our version of this same system is at work in the developing neural tube and will be why some cells will become a neuron and they'll relegate their neighboring cells to become a glial cell instead. Okay. So again, this is an ancient mechanism. We've been doing it this way a long time. Question about that? One thing that's not in the book, it will be in the second edition, it'll come out too late for you. Each chapter I've ended with a feature called the cutting edge. And I just wanted to tell you, because I think it's cool, about that, which is a recent experiment where they asked whether this same organizer business is going on in mammals, right? So they made a bunch of human cells and eventually treated them in such a way so that these cells would start expressing, right, and releasing an organizer. And how ancient are these mechanisms? Would a human cell expressing the human version of the organizer, would it replicate Hilda Mangold's experiment? And the answer is yes. So they label the human cells with something called tomato red, and they took the one that was expressing the organizer and put it in a weird place in the chick embryo, not at the chick dorsal lip of the blastopore, but in another place, just like Hilda did. And lo and behold, another nervous system started to develop in this embryo right where the tomato red cells were. And just as Hilda's graft of the dorsal lip of the blastopore in another location, just as those cells recruited, or if you like, exploited the native cells, these tomato red cells, human cells, organized the chick cells to start making a new neuraxin. Odds are then that we got here the same way. Okay. Let's step back. So what we've seen with conditional specification of cell fate, it's also sort of positional specification of cell fate. What you're going to be depends on where you are. And Sidney Brenner thought about the guy who came up with C. elegans and perfectly acknowledged that C. elegans do it differently than we do. He called this the European plan and the American plan. So the European plan is, your life is determined for the most part by who your parents were. Just like in the worms, your fate depends on who mom, grandma, great-grandmom, etc. were, right? And it's specified. So think of a guy who's born the third Earl of Canterbury or whatever, right? Of Cranberry, I like that, right? His fate is set right there, right? Just where he was born. And for thousands of years, certainly hundreds of years in Europe, the people who got the best resources were those who happened to be born in the right place. The American plan, and I must say, this is the ideal American. He said, in America, anybody can grow up to be president, right? I don't have to say any boy grow up to be president, right? Anyone can, and it's not a matter of, who your parents were. It's a matter of where you find yourself. You get yourself in an environment where you can go to school and learn to work hard or whatever. You can eventually change yourself. How's that? So this conditional specification of cell fate is a lot more democratic, for lack of a better word, small d, democratic. And again, what this means is, in conjoined twins like this, there's never any cell that's out of place. I mean, yes, it's unusual that there are two heads joining there, but for any individual cell, when you zero in on it, its neighbors are totally correct for where it is, and it's doing exactly the right job for its location, its position in the body. In other words, you decide what to become by noting where you are. And next, we'll get there today, we're going to work out how cells know where they are and therefore which fate they take on, again in fruit flies. But first, a Kahoot. Yep, here we go. More questions, because those were all great questions while you're logging in.
What?
And relevant to what Hannah was saying about how MCAT preparedness courses cost a lot of money, there's another case where in the American system, the affluent benefit more than, right? They'll get a head up, a boost up. What's interesting about standardized tests, I don't know if you've been following this, but there have been several elite schools who said, oh, you don't have to turn in SAT scores or ACT scores anymore. MIT, Harvard, places like that, where, because they were concerned that rich people are more likely to get those prep courses and therefore more likely to do well. And, it gives them a leg up. And indeed, as they looked at their past data, it turned out that if your family was in the top 1%, even if you had all the same scores as everybody else, you were like 10 times more likely to get admitted. So they got rid of the standardized test requirement. And then after a few years, they looked and what they noticed is there were fewer poor kids showing up in their class. So it turned out that there was a set of kids out there from lower socioeconomic status who, when they took the standardized tests, had a better chance of getting into those places. Expect more about all this influx. We'll actually talk about standardized tests at the end of the course.
I did so. They can all out what? Yeah, like.
Bye.
Yep, we call it induction, which is a particular kind of conditional specification of cell fate. Because of differences in pigment. The donor dorsal lip of the blastophore came from a frog that was pigmented, and then she implanted it into a frog that was albino, or vice versa. I forget. I think she did both. So in that case, and nobody had heard of fluorescent markers yet. Here's a tough one. It's enhancer of split. They found the gene because they noticed that mutations in that gene sometimes caused hairs to be forked instead of not. And the gene enhanced that. Got nothing to do with the function we're talking about, but is another instance of pleiotropy. Yep, noggin is one of the three. So delta is not an organizer. It is involved in the delta notch system of having cells determine which is going to continue on a neural fate. Okay. Okay. And I'll remind you, the name of that receptor is the TGF-beta receptor, but it is, BMP is indeed the ligand. Not much change. Okay. Last chance. Yep, they were red because they'd been labeled with, they were expressing the fluorescent protein, tomato red. Who's number 3? Shreya, where are you? Oh, thank you. Oh, well, okay. You wrote trust by Aiden? Who's number 2? Eli. Excellent. Number 1? I don't know your name. Emily. Okay. Oh, see, I see how consistent I am? Just one seat short of where I need to be. Story of my life. Ready for more? You're going to get it whether you are or not, of course. So here are the learning objectives. And now we've figured out what induction is, how one cell can communicate with another to change its gene expression, therefore change its developmental fate. And we can see how, in principle at least, all of that could, each cell telling the other what to do, could eventually end up with an organism with all the right cells in all the right places. And now we're going to deal with the question of how to put that together and form a body plan. And this is a very famous illustration of a very interesting fact, which is no matter which species, which vertebrate species you're looking at, early vertebrate embryos all look a lot alike. And yet, when they grow up, they're going to look really different from one another. And Darwin noticed this. And Darwin, I mean, he was well aware of these sketches and of this similarity. Actually been told by an American scientist before Haeckel published his book. And as he pointed out, if an adaptive trait if a trait isn't adaptive until later stages of development, there's no reason for the trait to show up sooner. And he noticed things like in pigeon fanciers, people who breed pigeons to have different structures and behavior. He noticed that the squabs, when they were first born, they all look alike. And that the new feature, the one that the breeder chose, doesn't appear until later in development. Which indeed what matters, because the breeder will look at the adult to decide whether to breed or not. And so everyone understand that for a new adaptation to arise and spread, you don't need to use it. It doesn't need to appear until you need it. Think about this, teeth in mammals. How come most mammals are born without teeth? Well, because they don't need them, right? The teeth are going to be important later. So why bother making the teeth early when they're not going to make any difference to the survival of the individual until later, right? Same with claws, right? Most mammals that are going to have claws as adults, they don't have them when they're young. In fact, it might be maladaptive for them to have those. But that's okay. They get the adaptive advantage when they appear in adulthood. There's another reason why all vertebrate embryos look so much alike. And that's the fact that any mutation that mucks about with things early in the process are not as likely to be survivable. You don't mess with the beginning, because any little change early in development is likely to lead to a cascade of changes that is going to be catastrophic, and that embryo will be lost. And I like to call the analogy to a donut shop. You can go into any donut shop, and there's lots of different kinds of donuts that are out there, right? Now, I'm going to pretend that there's only one formula for dough. In fact, okay, there's raised donuts and cake. Don't forget that. But you've got one dough, and the dough is always the same amount of yeast, same amount of sugar, same amount of flour, eggs, water, whatever it is, right? And yet, even though they all start out with the same dough, you can make them incredibly different from one another, depending on what you do in the later stages of donut making. Everyone get that? And in fact, if you mess with that early formula, if you decide, I'm going to leave yeast out this time, well, you're going to be out of luck. And in general, any little change early in development is going to have a cascading effect, and therefore is likely to have lots of different effects. And sooner or later, one of them is likely to be bad. And so another way to think of this is that most of the mutations that are responsible for the radiation of species, for one species being different from another, most of those mutations are ones that act late in development. Because you can have a mutation that acts late in development, and it's not as likely to lead to trouble with other parts of development, because the rest of the development already took place. Everyone see that? So this gets us to Haeckel's famous dictum that you've heard, or suggestion, that ontogeny recapitulates phylogeny. Because in Haeckel's mind, he actually thought that all these vertebrate embryos, even though some of them are going to make people, and some of them are going to make pigs, etc., he actually thought that all of them first made the body of a fish. And he said that because he knew that fish were the oldest group of vertebrate species out there. And this was sort of, what's interesting, there's this 19th century, actually 18th and 17th century German tradition of extreme romanticism, of seeing the world as being not just a mysterious place, but a wonderful, right? It makes for great poetry and novels, but it might not have been so good for science. And Haeckel, he was German, he wrote in German. His book was published in English, and it was an immediate bestseller. He had several editions. The world ate this up. And he loved Darwin, right? And Darwin was a little bit embarrassed about his over-exuberance, in fact. So in Haeckel's mind, this was like part of some grand plan, that we would all start off as fish, and then we'd become lizards, and then we'd become birds, etc. Then we become lower mammals, and then we'd become lower primates, and then we'd become upper primates, and everything is as it should be. And note, this puts us at the top of the pyramid, right? A very common idea in human thinking. You know, every time my brain tells me it's the most wonderful organ, I remind myself who's telling me. So when people are saying we're the acme of evolution, take it with a grain of salt. And so he proposed that ontogeny, the process of growing up for an individual, is recapitulating phylogeny, the evolution of different species on this planet. And that idea is demonstrably stupid. Even though you've all heard it. It's clearly not the case. So, and there's a little bit of a problem that he pushed that so hard, because you can find on the web, you can find folks who are evolution deniers, creationists, who claim that the whole idea of evolution is based on Haeckel's lie, right? And there's a particular reason for that. In the first editions of these illustrations, there were some embryos that got duplicated, and it's clear that was just a printer's mistake, right? And in later editions, he corrected that. But you won't read that on these, anti-evolution websites. You see, he was lying to convince people about evolution, which is crazy on several things. He wasn't lying. And number two, he had nothing to do with convincing people about evolution. Trust me. Nobody was paying much attention to him except lay readers, right? Not the scientists. And it turns out that when people have gone back to try to recreate that using his materials, it turns out that this similarity across the various embryos, Haeckel kind of stretched it some, presumably an honest mistake. As a guy who saw a little man inside the head of a sperm, he was probably seeing what he expected to see and sketching what he expected to see, right? Something that we're all guilty of. And the point I'm trying to make here is that when you look at mammals like us, these structures here, they never formed gills. In his mind, they were gills, but they're not forming gills. For one thing, you don't need any gills. In the fish, it will only be later changes in those bumps that will go in a different pathway and form gills, etc. So we call these arches, the pharyngeal arches, and they're, they differ from one vertebrate species to another, one vertebrate class to another. But there's no denying there's an overall similarity there. How's that so far? So no, ontogeny does not recapitulate phylogeny, and the theory of evolution by natural selection does not require that ontogeny recapitulate phylogeny. In fact, that would be impossible. Why should a mammalian embryo make gills at any stage of development? We're not going to live in water. Okay, enough. Okay, agreed. We're going to talk quite a bit about fruit flies, and I will say that of all the species that have been studied, I don't think it's a stretch to say most of what we learned about gene expression and development was first learned in flies, right? It's amazing what those guys accomplished. People like Thomas Hunt Morgan, and others who early in the 20th century realized that this was an ideal organism for studying genetics. And there are several advantages of fruit flies. Number one, even though they're annoying and we all hate them and want to kill them, they don't communicate any human disease, right? So you can kill them if you want to, but if you eat one, nothing's going to happen to you. And odds are you've all eaten some. They're everywhere. There are lots of different species. It was eventually he settles on Drosophila melanogaster. And among the things that's cool about them is that they have a reproductive cycle of only about 5 days. So you put a couple of fruit flies together today and you're going to have, another set of adults in just a week. So studying changes across generations becomes easier. And then there's other things about them that seem almost weird, right? One is they're exquisitely sensitive to ether, which was already available. So you can take a vial of fruit flies, expose them to ether, and they'll be anesthetized for a short while. And you can look at them under a dissection microscope, and it's easy to tell the boys from the girls because the guys have these little mechanisms in their genitalia to latch on to the girls. It gets better than that. As they go through development and become pupa and get to the adulthood, the last thing the adult does is eat the pupal skin that was in and send it down the digestive tract. And that material is dark. Pupa are darker, right? And it turns out if you anesthetize the female and flip her over, and you can still see that dark tract that's going through her gut, which you can see through her belly, that means she's still a virgin because she won't be mature enough to mate and to reproduce until she has passed that pupal skin out of her gut. Who could arrange that, right? So pretty cool. So they worked all this stuff out. Good. Why is it? Because it means that if you've got a jar full of flies, both sexes, and you anesthetize them, great question. You turn them all over. These are the boys. You use a hairbrush a painting brush to sort them. And here's some females. This female, she's still got the pupal skin in her gut. And I know that means she hasn't mated yet. Now, if I mate her with another animal, I know who the father is. Because that's the only father that's been available to her when she was reproductively competent. So cool. If I had it to do over, I might have done fruit flies instead of mammals. Yes, good.
So, you have to begin them.
What I mean, it's just a coincidence, but it turns out that she won't be reproductively competent and she won't mate until after the pupal skin. It's not that the pupal skin is inhibiting anything, right? It's just that's how the timing works out. And they're easy to grow. They worked out, making agar. They eat yeast. When you see them on your fruit, they're not eating the fruit. They're eating the yeast that's all around your fruit, right? And so you put yeast in the agar and you can buy yeast at any store, right? Yeah.
Should be an indicator for the male.
There's no such indicator for the males, but you don't need one, right? If you put him in with a virgin female, the offspring have to be his, right? He's the only one you put in there. Oh, and they worked out how to put them in these little dairy containers. They might get cream for your coffee. Or how to put the agar in the dairy container, put a little cardboard top, so all this got worked like 1910 or something. And when he left the East Coast and went to Caltech, it was a big part of what made Caltech famous. Yeah. And anyone who wanted to study genetics, you had to go to Caltech. Okay, Harvard, but those are the two places. You okay? And so people were very interested in, Morgan was interested in studying the genetic. He's the one who worked out how to tell how far apart two genes are in the chromosome, right? They worked all that out with mathematics and nobody knew what DNA was, right? But it was all tour de force, the stuff they worked out. Good thing you got a Nobel Prize. Being interested in ontogeny, development, people were looking at early embryos and realized that there was a fate map on the surface of the embryos. They got dyes that German chemists had worked out in order to dominate the textile industry worldwide. They found that some of these dyes were not that toxic. And so they could take a tiny little brush and put some colored dye on it and take a Drosophila embryo and brush a little bit of color in one place and then wait to see the embryo would develop. And since the vital dye didn't kill the cells, they would develop it. And what they learned is, and we won't talk about how they knew where they were, notice there is an asymmetry in the egg, right? There's an end that's more pointed and an end that's not. And so they eventually worked out that this part of the animal, this part of the embryo was going to make the head. And this part of the embryo was going to make the nervous system. And this part was going to make the brain and the mesoderm and the legs, etc. And so they had these things called fate. So they knew that if you look at the embryo and you know which way to orient it, they knew that certain cells were eventually their offspring were going to divide and form certain structures. But they had no idea how that got specified. And so it'll be in the late 20th century that the fruit fly people figure that out. And there's some specifics about fruit flies that I have to tell you to understand what they accomplished and how they did it. So unlike mammalian embryos, gastrulation is very different in fruit flies than it is that we've talked about so far. And I'm not going to talk about that in detail because we don't need to. But there's a weird thing about the egg in Drosophila, which is instead of the cell dividing, it doesn't. It stays, that egg, which you can see with your naked eye, it stays one great big cell with one cytoplasm for quite a while. Well, how do you divide then? Well, fruit flies, it's very efficient. The nuclei divide, but they don't bother putting up cell walls between them. And so there reaches a point where you have an embryo that is only one big cell, one cytoplasm, continuous with everyone, but it's populated with nuclei, and they are spread throughout it, and they're all distributed pretty much evenly. So we have a name for that. We call it a syncytium. And that cell, while it looks mostly like we can see some asymmetry in its shape, mom, once again, has arranged for it not to be the same everywhere. So here's an example that's well known about the distribution of a maternal factor. And this one happens to be for, which is it here? Nanos. I forget if this is Nanos and Bicoid. Anyway, and so it turns out when mom made the egg, she made these anchor proteins that are more concentrated at one end than the other. And they have attached to them mRNAs for a transcription factor. So that's how she arranged for the cells at this end of the embryo to take on a different fate than the cells at that end of the embryo. One of the things that's happening, so there's all these nuclei in there, and they're all transcribing. They're all expressing gene. But notice that they can influence each other's gene expression without the bother membrane signal, because there is no membrane between them, right? So the first thing that starts happening is there's this flurry of expression of transcription factors, and once one nucleus makes a transcription factor protein, it's free to interact with the neighboring nuclei and affect them, right? It's a very efficient way of working out who's going to do what. And it turns out that there's some genes called maternal factor genes, some of which are right away expressed a lot in the back end, the the caudal end of the animal and some at the head end. So bicoid is nanos is expressed at the caudal end and bicoid at the front end. And here's an example of how you can see that expression. So these are only nuclei, but we can still do in situ hybridization to keep track of where particular mRNAs are being made. And there's a lot of bicoid being expressed in what we know is going to become the anterior end of the animal, and almost none at the posterior end. And we're not showing it here, but Nanos protein is being similarly concentrated at the opposite end. Once people realize this, and by the way, both bicoid and nanos, they're both transcription factors. So they bind to DNA and affect gene expression. Once people realized this, they did experiments the Drosophila way, which is they made mutants to see what would happen. Bless you. And a very interesting thing happened, which is that so in wild type embryos, bicoid is expressed at the anterior end. And as normal development continues, you can keep track of various body structures that are starting to form at various places along the neuraxis from head to tail. Everyone get that? And what they did was they made some mutants where they had disabled the bicoid gene. So the bicoid gene, when we put negative there, that means it's not producing any functional product. Either they knocked it out or they messed with it such as dysfunctional, a so-called knockout. And when they did that, of course, when they did that, no viable embryos came out, right? So that shows that bicoid's crucial, right? But what was interesting was they noticed that the products that tend to be found at the tail were now being expressed at the head end. Wait a minute. This is always supposed to be, so they ended up making an animal that had two tails, and the embryo was doomed, didn't get very far, but they thought, well, so maybe bicoid's important for specifying the head end. And so they did experiments like this where they, in bicoid knockout embryos, they added some bicoid mRNA. And if they put the bicoid mRNA at the anterior end and waited, a normal larva was made. So that proves that bicoid is necessary and sufficient to specify the head end of the larva. And you say, well, maybe all it's doing is it's, putting the mRNA there. You just messed with things a little bit and happened to correct a problem. But if they put the mRNA in the middle of the organism, in the middle of the embryo, lo and behold, the only structures that went down the pathway of differentiation be the head were in the middle. Another doomed embryo. Or they could even add bicoid mRNA to the back end of a wild type embryo. And when that happened, they ended up with two heads. So this maternal polarity gene that mom put at the anterior end of the head was enough to trigger the proper orientation of the whole animal. How's that? Well, it turned out there are several maternal polarity genes. Bicoid and nanos are among them. And they're called that because perturbations of those genes end up messing with the anterior-posterior axis. And they're all genes they're all transcripts or proteins that are unevenly distributed in the egg so that normal development takes place. And what's interesting is that in the case of bicoid and nanos, I don't remember if this is nanos or another maternal factor gene, they have opposite effects on each other. That one of the things bicoid does is suppress the expression of Nanos. And one of the things Nanos does is suppress the expression of Bicoid. And so if there's a little more Bicoid at the beginning, at the anterior end of the cell, that means that it's going to suppress expression of the posterior product, right? So you end up with this sharpening where presence of anterior factors in the front end suppress the expression of caudal factors and vice versa. And then at a later stage, the embryo doesn't stay a syncytium forever, but at a later stage, cellularization takes place. They all each nucleus starts making cell walls around each other. And by the time that happens, there are already some cells that are fated to make the head, some the thorax, some the abdomen, and some the tail. So there's this mutual antagonism between them that ends up sharpening and strengthening the spectrum there. And that's good because you don't need two heads, right? You really want one head, one tail, and all the stuff in between. And that happens because these transcription factors are affecting each other's expression. Well, it turned out that in addition to the maternal polarity gene, here we are doing that some more. So that's just further another way of mapping it out. What's going to happen now is this continuum of expression of the maternal polarity genes. One of the things those products are going to do, their transcription factors, now they're going to affect which genes those nuclei express next. And if you look, you could think about it from back here, every cell in there is getting you a unique combination of the two transcription factors. And so which transcription factor they make next depends on what particular combination of Bicoid and Nanos they had. Everyone see that? What we're going to see next then is there is a cascade of effect, a hierarchy of gene products, mostly transcription factors, that are going to affect transcription at later dates. And as development continues, there's going to be this unfolding of different patterns of gene expression. And that's going to arrange for a particular pattern of body development. Okay, so far? Good. Just
It's like, say, like a leg and the front of the insect and a leg at the back.
right.
Do the cells have to migrate? Like, are they in some center?
It's a great question. At a later stage, there will indeed be lots of migration going on, but we're not there yet. And the migration, of course, is also a response to genetic instructions, which is to say what genes they're expressing, right?
Sure.
So there is here's another way of looking at it. Here's Bicoid being expressed mostly in the anterior end, Nanos mostly at the posterior. And because they are interacting with each other, there's a second layer of transcription factors called hunchback, whose expression is going to be controlled by the combination of those two genes. Done.
Wondering, is the gradient the most important part, or is it the concentrate gene?
Well, let's see. I would say the most important part is the gradient, because it means that each nucleus in the syncytium is getting a slightly different combination of the transcription factors.
Like, there's a certain point where if you lowered the concentration but staying the gradient, like a head would go at least they have to grade. Like,
well, I mean, I don't think you can have a gradient unless there's a concentration at one end or the other, right?
the concentrate.
Presumably, if you lowered the concentration of any one gene, it would depend. If you lowered the concentration of Bicoid, you'd end up with less head. And if you lowered the concentration of Nanos, you'd end up with more head and less tail. And And fruit fly people being what they are, people did those sorts of mutations. You can be sure, right? Any Byzantine combination would be possible. Anyway, so what's going to happen now is the differential expression of these two antagonistic transcription factors is going to affect the transcription of yet another gene, in this case, hunchback. And the rules of transcription factors are such that at this part where there's lots of bicoid and no nanos, they're going to express a lot of hunchback. And in fact, there's even this little bump there. And then as you get to parts that are being exposed to more and more nanos and less bicoid, the amount of hunchback transcription goes down. Hunchback is one of a class of genes that are known as gap genes. So the maternal gene, the maternal factor genes, are going to control the expression of the gap gene. And you can be sure the gap genes are going to control the expression of another hierarchy of genes that are going to be expressed later in development. So at the earliest stages of development, only the maternal factor genes are being expressed, and there's only a few of them. I forget, 6, 7, 8, something like that. But they're going to determine the expression of the gap genes. And there are more gap genes than there are maternal factor genes. And in fact, the gap genes are going to be expressed in fewer parts of the body than the maternal factor genes. The maternal factor genes, each one is expressed in half the body, right? Basically. And so you'll end up eventually in a situation where, like this, where if we do the in situ, there's yet another set of genes, the pair rule genes. So the maternal factor genes control expression of the gap gene, and the gap genes control expression of the pair rule gene. And by the time you get to the pair rule genes, each one is being expressed in a very circumscribed portion of the embryo. So that here, there are two different pair rule genes here. One's being shown with a red marker and one with a black. And look how beautiful that alternation is. And by the way, notice how sharp the lines are. This nucleus is expressing, right, Fujitaratsu, I think, and this one's expressing the other. And there's very few cells that aren't expressing one or the other. Another clue that there's probably some sort of interaction going on, these transcription factors affecting each other's expression, because that's how you'd end up with that sharp demarcation. So wait a minute. So the maternal factor genes were expressed basically in half the embryo. The gap genes were expressed in a smaller portion of the embryo. And by the time we get to the pair rule genes, each one is being expressed in a very specific portion of the embryo. And also notice that for even skipped, right, there's 1, 2, 3, 4, 5, 6 different places that are expressing 6 different demarcations. They're expressing it. And for Fujitaratsu, 1, 2, 3, 4, 5, 6, 7 different regions, they're expressing that one. So already we've started divvying up the embryo. And this divvying up the embryo is going to result in them making different parts of the fly.
Do the gap genes control the pair rule gene?
Exactly. Do the gap genes? Yes. And so the hierarchy continues, where a small number of maternal factor genes control a slightly larger number of gap genes, which control the expression of a larger number of pair rule genes. You can already see where this might be going, right? Because there's going to be another hierarchy. And with each of those steps, more and more portions of the embryo are getting demarcated and their fate being determined. So, the gap genes control expression of the pair rule genes, and they affect the expression of the segment-polarity gene. And these are various examples of them. So here's hunchback, right? which is a gap gene, and it's the one that's being expressed in orange there. And you read over here, you can see that these other colors represent other gap genes. Look how different their expression patterns are.
Oh.
So the gap genes are being expressed in large portions of the embryo. Pair rule genes are being expressed in a smaller portion of the embryo, and the segment polarity genes are being expressed in yet another smaller portion of the embryo. And notice that the number of domains being expressed by each of the genes also goes up, right? This segment polarity gene has a lot more than 7 regions where it's being expressed. One of the things that's happening here, by the way, is these cells that are expressing this particular segment polarity gene called engrailed, these cells in this portion of the organism, right now they're both expressing engrailed versus that portion, but notice that these cells have a different history of transcription factors that they've been expressing than these cells. Everyone see that? Because these cells had a different, either they were or weren't expressing the pair rule gene that controlled them. So even though, here we're just keeping track of this particular gene and it's being expressed in these different segments, but the different rings, but these rings are not equivalent. Even though they're all expressing this particular gene right now, they have a different developmental history of what transcription factors they were exposed to in the past. And so, in fact, the cell fate of these cells is different from these cells and these cells. For one thing, for most of these cells, they're going to end up making different segments, or being the nervous system, making different segments, and we've already worked out how each of the segments will have a different nervous system. So here's the way things are working. There's a maternal polarity gene that are controlling the gap genes. They're comparing the pair gene that are controlling the segment polarity genes. And eventually there's going to be a set of cells that are going to make particular structures. The cells in this portion are going to make the head. The cells in this portion are going to make the tail. They're going to make the abdomen. These cells are going to make the thorax, etc. And it's a hierarchical in every sense. The maternal polarity genes, they have to come first, right? And also, there aren't that many of them, right? There are a lot more soldiers than there are generals, right? So it's hierarchical in that sense too. Also, there's a set of control. The highest part of the hierarchy, the maternal polarity genes, they have the most influence. Now then, Allison.
I think the next class starts really soon.
Oh my God, Stigi. Thank you. Oh,